This is the second video in which we will look at the adaptations induced by strength training. For our outline, we'll first look at the time course and signaling events leading to muscle growth. Then we'll look at what happens to muscle after a period of, or during a period of detraining that follows a period of strength training. And finally, we'll look at this interesting phenomena in which athletes are concurrently doing strength and endurance training, and we'll see how that affects the muscle. So here's some questions for this lecture. You can see the pages are 318 to 324. And our learning objectives will be these. How does resistance training affect muscle protein synthesis? What's the master regulator of exercise-induced protein synthesis? I'll give you a spoiler alert here. It's mTOR. How do satellite cells contribute to hypertrophy? What are the effects of detraining on muscle strength and fiber size? And why does concurrent endurance and strength training inhibit strength gains? We'll start with figure 13.17, but before we get into the details, let's first address muscle hypertrophy. So muscle fibers get bigger with the course of endurance training, and the reason for this is because there is an increase in the number of, or the amount of, contractile proteins within the muscle fiber. This occurs because there is an increase in muscle protein synthesis that is greater than muscle protein degradation. It turns out both of those processes will increase after a resistance training bout. So muscle protein synthesis will increase, but also muscle protein degradation will increase. The reason it takes a while before fiber hypertrophy can be measured or noticed is that the muscle protein synthesis has to be greater than the degradation rate for quite some time, several weeks, before we will be able to measure this. So let's go back to figure 13.17. This shows us on our x-axis we have the time in hours post-resistance exercise, so the time after you've completed your exercise bout. And note that it's 24 hours would be 1 day, is about right there. And on our y-axis we have the percent increase in muscle protein synthesis. So this can be measured by you give a subject a set of amino acids that have been marked. Say some of the carbons are C13 instead of C12, and so then we can follow these after some period of time when you can take a muscle biopsy and you see how much of this how much of these labeled amino acids have been incorporated into the muscle. And so we can see that the blue line would be a trained subject, and the red line is the untrained. The peak response is about the same. You get this about 150% increase in the rate of muscle protein synthesis. But then with the trained person, it declines rapidly after about 6 or 8 hours, maybe 10 hours, whereas the untrained person stays elevated for a longer period of time. If you think about our big picture going back to the mRNA levels and the protein synthesis that's going to occur after an exercise bout, this sort of makes sense, right? To the trained person in this figure has experienced a lesser challenge to homeostasis than the untrained person did, and so the response is going to be slightly less. And so this is an example, again, of the overload principle. You keep having to do a greater and greater stimulus to get the same gains. So now that we see what happens with each exercise bout regarding muscle protein synthesis, you can think about what happens over the long term. If you're doing a strength training exercise bout 2 to 3 times a week over the course of 6 or 8 or 10 weeks, you can imagine then that you are continually increasing the muscle protein synthesis in your muscle fibers, and therefore they will slowly build up more contractile proteins and causing hypertrophy. So what are the signaling events that cause that to happen? Well, let's take a look at the next figure. This is figure 13.18, and this shows the signaling pathway that ultimately ends up in creating muscle hypertrophy. As you can see in this figure, the master regulator for protein synthesis is called mTOR. It's right here in the pathway. Downstream from mTOR then is an increase in protein synthesis. This molecule primarily works by increasing the translation step, and so that is the process that takes place at the ribosomes in which the mRNA sequence is then translated into a peptide sequence. And mTOR, I should point out, stands for the mechanistic target of rapamycin. Sometimes you'll see that called a mammalian target of rapamycin. I've seen it both ways. mTOR is activated by two molecules. One is PA, or phosphatidic acid, and the other one is called Reb. All three of these molecules, PA, Reb, and mTOR, are associated with a lysosomal complex, and our figure here just calls it the lysosome, and this is in the cytoplasm. So now let's take a look at how mTOR is activated. At rest, mTOR is inactive because PA levels are quite low, and Reb is actually being inhibited by another molecule called TSC2. So to activate mTOR, we need to either increase the concentration of PA, or we need to remove the inhibition on Reb. It turns out with muscle contractions, both of those events happen. Let's now go up to the top of this pathway, and we can see that the main primary signal for this pathway is a mechanoreceptor activation, and that's shown here. So this is a mechanoreceptor that's sitting in the sarcolemma, and so when we're doing our strength training exercise, we're creating tension in the muscle and some stretch along the sarcolemma that activates that receptor. It then will enhance PA synthesis, shown here, which then of course increases your PA concentration. It also activates a kinase called ERK, and that inhibits TSC2, and you can see the straight line there instead of the arrow in front of that box. That indicates that's an inhibition, and so if we inhibit TSC2, we've removed the inhibition here on Reb. So by activating the mechanoreceptor, we end up getting an increase in phosphatidic acid and an activation of Reb, both of which will enhance mTOR activation and then the downstream effects of enhancing protein synthesis. So that covers the signaling events that occur that lead to protein synthesis and then ultimately muscle hypertrophy. But we can't ignore what else is also necessary, and that is, of course, the amino acids. So nutrition is important here. The muscle cell has to have available all of the necessary amino acids to build these proteins, which means especially the essential amino acids. There's been lots of research on this topic over the last few years, and the studies seem to show that if you take amino acids or just eat a regular meal and provide the muscle fibers with an increase in the concentration of those essential amino acids, then you will increase muscle protein synthesis. And this is most enhanced either if you eat right before your strength training exercise or within an hour or two after. To look at the time course of these events, let's go to the next slide. This is figure 13.19. You can see that Reb and PA are activated within seconds, mTOR is activated within minutes, and then over the next few hours, and maybe even lasting up to 24 hours, you'll see your increase in protein synthesis. Those proteins will then accumulate day after day after day and ultimately result in muscle fiber hypertrophy. We know that this hypertrophy can occur for weeks and months and maybe even years, but ultimately it will be limited by the activity of each nucleus. So each nucleus can only produce so much mRNA that can be made into protein. So one way muscle gets around this is by actually increasing its number of nuclei. So these end up being called myonuclei. And this is shown in figure 13.20. So where does muscle get these extra nuclei? Most cells can do this by simply copying the DNA in the nucleus and then splitting, and that's typically how mitosis would occur. But in skeletal muscle, the nuclei do not do this. It actually incorporates satellite cells that are sitting right outside the sarcolemma and internalizes them, and then they become new myonuclei. So a satellite cell is an adult stem cell. It sits right outside the sarcolemma underneath the connective tissue, and it's activated by activity, so strength training, as well as muscle injury. When these satellite cells are activated, they fuse with the muscle fiber, they become internalized, and then they too can start producing more mRNA to make new proteins. So by doing this, the muscle fiber, as it gets bigger, will be able to keep producing proteins as its cytoplasmic volume increases. There's evidence to show that this is a pretty robust response in younger people. We can easily activate and incorporate satellite cells into our growing muscles. But this ability decreases as we get older. So the ability for 80-year-olds and 90-year-olds to accomplish muscle hypertrophy is actually decreased, probably because of a reduced satellite cell activation. So that covers our mechanisms and time course of muscle hypertrophy. So next, let's take a look at what happens to muscle when we stop exercise training and go through a period of detraining. This is shown in figure 13.21. In this study, subjects were trained for 20 weeks and then detrained for 30 weeks. I'll write it down here, 30 weeks, and then they did a 6-week period of retraining. And so let's see what happens. In panel A, this shows the maximum strength, and panel B shows muscle fiber size. So let's look at panel A. We can see that with 30 weeks of detraining, the subjects lost 31% of their strength. But in only 6 weeks, recovered basically all of it back. So they're almost back to their 100% level. In panel B, we can see what happened to the specific fiber types, at least to their cross-sectional area. Type 1 is in orange, and we can see that 30 weeks of detraining, we only saw about a 2% decrease in size, and then the retraining maybe slightly larger, but not very much change in type 1 fibers. And we wouldn't necessarily expect this because type 1 fibers have been shown to not respond very robustly to strength training. The type 2 fibers, on the other hand, we can see in the green, the type 2A, we had a 10% decrease, and then an increase again back up above even the initial train state with the retraining, and a similar response in the type 2X, so a 14% decrease and then another increase with retraining. So we can see the type 2 fibers showed a greater atrophy and then a greater hypertrophy than the type 1 fibers. But also note the percentages here. We saw a 31% decrease in the strength, but only a 10 to 15% decrease in the muscle size. So that indicates that this strength loss was due only a little bit to loss of muscle mass and was probably then primarily due to neural mechanisms. Another piece of evidence for the importance of the neural mechanisms is that during the retraining phase, which only lasted 6 weeks, we can see that the subjects got almost all their strength back, and we know that neural mechanisms can occur fairly rapidly, while hypertrophy tends to be delayed. The change in muscle cross-sectional area, the fiber cross-sectional area shown in panel B here, is an interesting one because think about the magnitude of the change. So 7 months, this is 30 weeks, so this is 7.5, 8 months of detraining results in only a 10% loss in muscle fiber size. And so this is an interesting difference in what we would consider, or what we would call structural proteins, these sarcomeric proteins, versus the metabolic proteins, the enzymes involved in our metabolic pathways. Recall that with oxidative capacity, if the person stopped endurance training, they would lose 50% of the gain that took 5 or 6 weeks to gain. They would lose it in 1 week, right? And then you could get it back in about 4 weeks. But the rapidity of that is much greater than the changes you see with these contractile proteins and muscle size. Now let's turn to our final topic, which is the exploration of what happens to our muscles when we do strength training and endurance training concurrently. It was shown back in the early 1980s that when subjects were engaged in both strength training and endurance training, that the gains in strength were less than the subjects who were only doing strength training. So there was something about the endurance training that was inhibiting the strength gains. over the years, there's been quite a bit of research into why this happens. And as our book points out, they put forth 4 general hypotheses. And the first one they call neural factors, and suggest that concurrent strength and endurance training somehow alters motor unit recruitment and therefore decreases strength. However, they then go on to say there is very little evidence for this, and so I think this is still a theory, so I'm going to sort of cross that one out and give it a big maybe, if not a downright thumbs down on this one, because we just don't know. The second potential mechanism for reducing strength while doing concurrent strength and endurance training is a low muscle glycogen content. The theory here would be that the endurance training will decrease your muscle glycogen content, and then in your subsequent strength training bouts, you would not have as much energy, and therefore you would be able to do fewer reps and fewer sets, and so you wouldn't create the large stimulus that you otherwise would get if you weren't doing endurance training. So this one, I don't know how much evidence we have for this one, but it's at least plausible, right? The third factor put forth is overtraining. That is, the muscles are not having enough time to recover between exercise bouts, and therefore they can't engage in this protein synthesis needed, and so the strength gains are compromised. Again, that paragraph points out that there's no evidence for this, and so to date, this is still just a best guess. Finally, the book puts forth the fourth mechanism, and this is the one that we actually do have evidence for. This is the depressed protein synthesis, and I'm just going to make a green check mark right now. And this is shown in figure 13.22. We can see on the left side of this figure that resistance training will increase your mechanoreceptor activation, which then will increase mTOR and your protein synthesis and muscle hypertrophy. On the right side of the figure, we see endurance training will increase AMPK activation, CAMK, CAM kinase, and P38, which will increase your PGC1 alpha and mitochondrial biogenesis. But also look at the green box here in the middle, TSC2. We saw this earlier in the activation of mTOR. Turns out TSC2 is activated by AMPK during endurance training, and we saw earlier that TSC2 is a negative regulator of mTOR. If TSC2 levels are elevated, or if it becomes more active, it decreases the activity of mTOR. And so what has been shown is that endurance training will increase your AMPK activity, increases TSC2 activity, which then decreases the activity of mTOR. So even if the person is doing strength training, mTOR has been inhibited, and so they won't get the protein synthesis response that subjects not doing endurance training would get. Whether or not this is the only mechanism that decreases strength gains while someone's doing endurance training is yet to be seen. So that concludes the coverage of all of the pages in the assigned reading for Lecture 35. But I wanted to leave you with one more thing to think about, and so I've added one more slide, and that is, why should we care about muscle hypertrophy? This is sort of the so what question. What's the importance? And so I put together 3 slides here and put them on this page. Of course, muscle growth, muscle strength, muscle hypertrophy are all big questions for many different areas. And one of them is livestock. This is the one on the left. Of course, if you're a rancher and you're raising cattle or pigs, you want them to gain the most weight, and preferably in muscle, with the same caloric intake. So this the cow that's shown there is actually called Belgian Blue. This is, or a double-muscled cow. These cows have been around for a couple hundred years, and farmers keep breeding them because, of course, they have enormous muscle mass. And recently, when molecular techniques were made available, someone did an analysis, and it turns out they have a mutation in one of the genes in muscle that limits muscle growth. It's called myostatin. And so now that the myostatin gene has been mutated, these muscles keep growing and growing and growing. And so you have these cows that look really muscular. I'm guessing they may not be that good to eat, though, because otherwise you'd see these cows everywhere. So my guess is their muscle is really lean and probably doesn't taste all that good. Another interest here is in the world of molecular biology and muscle research. And so when this myostatin gene was discovered, of course, then what did researchers do? They created a mouse that has the myostatin gene knocked out. And that's shown right here, this myostatin knockout mouse. And you can compare it to the normal mouse. You can see it's much bigger. And then look at the musculature here. This looks like Superman, a mouse that has done lots of weightlifting is what it looks like. So an enormous increase in hypertrophy. And that led to the question, does this happen in humans? And it turns out recently in Germany, the physicians noticed that this 7-month-old baby had really large muscles and for a baby of its size. And sure enough, they did a genetic analysis, and it has a mutation in his myostatin gene. And so he will have large muscles. We'll have to see over time what effect that has on his physical traits. And the final point I'll make here is I don't have a slide for it, but muscle strength and muscle hypertrophy as we age is more important all the time. We're learning more and more about muscle function with aging. And this is a big push in the medical community because, and it's of great interest because not only the money involved, but the debilitating lifestyle that many older people lead because of weak muscles. It turns out something like 80% of elderly people who live in nursing homes are there not because of mental lapses or mental deficiencies, but because of weak muscles. They cannot do the daily tasks of living, like cooking, walking up and down stairs, going to get the mail, cleaning, vacuuming, these sorts of things that we need to do to be able to live alone. And so understanding hypertrophy, muscle growth, especially as we age, is becoming a really hot topic of research. So that's the end of Lecture 35 and our discussion of strength training and muscle mass.